Dear viewers, Assalamu alaikum. Today we will discuss a case study on my PhD research work. In that study, we designed a drug and then we synthesized their structural analogues, and then by in vitro activity analysis, we also found structure activity relationship and ADMET studies. The research work was published in International Journal of Molecular Sciences. So let's start with the detail that how we designed, synthesized SAR and ADMET studies. So first of all, we should know what are the different approaches to drug design. So there are two different kind of approaches towards designing a drug. Number one, a structure or target-based drug design, in which we focus on the type of target or the active. According to that target, we design a drug that can fit or that can bind into that target. And the second case is the ligand-based drug design. In that kind of drug designing, we actually study the kind of compound or the compounds or clinically used medicines that are active against a specific target, and due to their structure activity relationship, we design our compound or drugs which are novel, and then we analyze their activity against the target. So first of all, our study was against carbonic anhydrases. So we should know what is carbonic anhydrase and the function of carbonic anhydrase in our body. It is a metalloenzyme, and it catalyzes the interconversion of carbon dioxide and bicarbonate ion in different parts of the body, like in tissue and lungs and kidneys, etc. There are 15 carbonic anhydrase isozymes, out of which 1, 2, 9, 4, and 12 are most important. And in this case, we studied 4 of the isozymes from human and then bacterial isozymes. So we know that it is a metalloenzyme and it contains zinc in the active site. There are some important clinically used carbonic anhydrase inhibitors. They inhibit carbonic anhydrase. So if we look at the structure of that compounds like acetazolamide, methazolamide, brinzolamide, and topiramate, we will observe that the structure which is common in all these is sulfonamide group. So if we look at the sulfonamide group, so this is the common group in all of these structures. It means that it has importance with the activity of the compound against that target. So based on this compound, which is SLC0111, it is human carbonic anhydrase 9 isozyme specific inhibitor, which is in clinical trial for the treatment of hypoxic tumors. If we look at the structure, there a sulfonamide group, benzene sulfonamide group, is the head, which is zinc binding group, and there is a urido spacer, and then a tail, which is a chlorobenzene. So in this way, this compound is our base compound, or we can say that if we are going to making a drug based on ligand, so we have taken this compound as a ligand, then we design our drugs according to this compound. So it is our standard or our compound. We are going to make different structure. So tail extension approach for novel carbonic anhydrase inhibitors. So taking that compound according to making that compound or reference compound, and we designed a compound in which we extended this tail. So if we look at the structure, this is our head group, which is zinc binding group, then a urido group, which is a spacer, and then this is a tail which is extended. So in both cases we have extended the tail, and the position of the substituent is varied in both cases. So in this way, this is our drug design, and then we design a synthetic scheme on the basis of which we can synthesize our compound. So it is based on different reactions by taking a precursor, which is a sulfanilamide or methanilamide. So these metanilamide, sorry. So sulfanilamide and metanyl sulfonamide group, then it is sulfanilamide, and if it is at meta position, then it is metanylamide. So by reacting with the phenyl chloroformate by esterification in presence of this conditions and solvent, it is converted into this compound, and then it is again treated with tertiary butyl 2-aminoethyl carbamate, and then reflux at 100 degrees centigrade, then we produces a Bog protection, and then we, with the help of fluoroacetate, a salt formation occurs, and then different benzaldehyde groups containing different R chain benzaldehyde, they are treated with this or salt, and then by the reduction with sodium borohydride we get this final compound. And you can watch this detail. You can visit my research paper to for detail of these reactions. So we get almost 16 to 25 we get 10 compounds from this structure and 10 compounds from this structure by making the different R groups by variation in R different functional groups. So here I am going to show you that the structural analogues of our compound. So we have a series 1 where it is sulfanilamide group and it is in para position, and from compound 16 to 25 we have different R chain at this ring position, and similarly with metanylamide we have 10 compounds from 26 to 35 with varying this R group. So the R group may be chloro, fluoro, or the position is also mentioned that 2 fluoro or 4 fluoro, and then hydroxo, nitro, and similarly bromo and hydroxo and phenyl and oxymethyl. So in this way, by changing the benzaldehyde, this benzaldehyde, we get the different structures or different analogues of these compounds, and finally we get 20 different structural analogues, and then we purified that compounds through flash column chromatography and recrystallization, and then characterized through proton NMR, carbon NMR, fluorine NMR if we have a fluoro group, and then mass spectrometry and melting point, etc. So after this purification and characterization, we have performed their activity analysis against carbonic for different human carbonic anhydrases. So it is in vitro activity, that is performed in the experimentally in the lab work. So we found that the percentage inhibition against different concentrations of our compounds against human carbonic anhydrase 2. So here we have only shown the data of these compounds against human carbonic anhydrase 2. So these are the compounds, and here is our standard acetazolamide. So if we look at the IC50 values plus minus standard error of mean, so we get these results. And how to find IC50 values from percentage inhibition? There is a video on my channel in which you can analyze, you can study that how we can calculate IC50 value by using GraphPad Prism. And there is also a video in which I have also mentioned that how we can find standard error of mean through bar So after finding this, we tabulated this table, and if we look at the values here, we have shown that the yellow highlighted represents excellent inhibition against human carbonic anhydrase 2, and we can also compare these results with standard drug, which is acetazolamide. So the value is 5. Here we have our compound which gives 2.8, IC50 value 5.3, 3.9 and 9.3. So these compounds are more active compounds, and then we can convert it into inhibition constant Ki value through kinetic studies. So for kinetic studies, we know that it is our competitive inhibitor. So these are the competitive inhibitors. So by following this equation, conversion of IC50 value to Ki, we use Chang-Prosov equation. So this is our equation which converts also, I have a video on this, that how we can convert IC50 to Ki value by using Chang-Prosov equation. So all the links of these videos which I have mentioned in this video, I will upload in the description box, so you can find that links. So I will make that mention in the description box. Then structure activity relationship, how we can find structure activity relationship for different compounds. Here we have data through in vitro analysis, the Ki values. By comparing the Ki values and the structure, we can analyze that what is the structure activity relationship. If we look at here, these are the sulfonylamides and these are the metanylamides, and the data is against human carbonic anhydrase 2. Then we look at the structure from here. It is 2-hydroxy 4-bromo or 2-hydroxy and 4-bromo. So if we look at the values, it is 5.3 nanomolar here and it is 2.8 nanomolar here. This means that the metanylamide position is more providing more activity as compared to sulfonylamide, that is para position. So if we look at here, the value is 3.9 nanomolar and 33.4 nanomolar, and in this case the orthofluoro is more active, but orthofluoro according to this, the para position or the sulfonylamide fluoro will give more activity, that is 3.9 nanomolar, which is more active than this one. So it means that this is more active due to this para position. And when we look at the structure, that again we have a fluoro compound which is more active, but in this case the fluoro is at para position. So this fluoro which is at para position, and according to this it is para urido, it means sulfonylamide, it is more active, that is 9.2 nanomolar, and here it is less active. But if we look at compare these two, then we will observe that from these two the orthofluoro is more active than parafluoro according to these values. So in this way we find structure activity relationship. Then extra crystallographic studies we performed for ligand target interaction, and we used synchrotron from France, and then we crystallized our compounds inside that enzyme, and by making a complex, and then we sent our sample to the lab in France, and then the crystallographic data was analyzed through different softwares, and then we observed that this sulfonamide group, amino group of the sulfonamide, will bind with zinc plus 2 ion, which is already bound with histidine residues, and then they will make hydrogen bonding with threonine 199, histidine 200, and protein 201 with this this one NH group. This is NH and this is NH. So it will make hydrogen bonding, and these are the hydrophobic interaction with that residues. This is our compound in cyan, which gives that the active site binding inside the active site of the carbonic anhydrase, human carbonic anhydrase 1. And for this, here is our data. Our data was submitted in protein data bank, and these are the code PDB codes, and you can visit that PDB codes, and then you can analyze that our compounds are crystallized inside the active site. And here we have in PDB protein data bank, which is a server, a database for different protein crystal structures, and you can visit that website and watch that structures. So after that, we have also performed molecular docking studies for ligand target interaction, but this time we have used human carbonic anhydrase 2 for ligand target interaction, and here we observed the same type of interactions with this zinc binding group, but the difference is this binding asparagine 67. Therefore, due to this binding, our compounds are, I think they are more active as compared to their activity against human carbonic anhydrase 1. So these are the specific amino acid residues which are only present in human carbonic anhydrase 2 but not in 1. If we look at here is the isozyme specific amino acid residues of carbon. Look at 67 position, all these amino acids are different in different isozymes, whereas in 62 only asparagine 62 is present in human carbonic anhydrase 2, 9 and 12, but in 1 it is valine. So therefore our compounds were more active against human carbonic anhydrase 2 as compared to 1, and they were active against 9 and 12 also, but the more specifically our compounds were active against human carbonic anhydrase 2. If we look at the structure activity relationship of our compounds based on in vitro analysis, X-ray crystallography and molecular docking studies, then we can observe that this is our head, which is zinc binding group, and it is due to this one, it will bind with the zinc inside the active site, and then this is our spacer, which is involved in interaction with specific amino acid residues. And then there is the tail with different functionality at the tail affects the activity of a compound by hydrophobic interactions. Okay, now this is our pharmacokinetic studies, our in silico ADMET prediction. We have also a video on how we can check ADMET profile, or how we can study pharmacokinetic properties through different softwares. So we have used different softwares, and then we studied the ADMET profile of our compounds. So here we have shown 18, 19 and 29 and 24 and 34. So these compounds which are active, most active, we studied their rule of 5, and for these physicochemical properties, that rule of 5, molar refractivity or log POW, we will make another video on physicochemical descriptors in our next video. So if you look at the rule of 5, they obey rule of 5. Yes, molar refractivity is 92 and 101, and then log POW is not very much high, which is 1.4 and 1.5, which gives that the compounds have good oral absorptivity. Then log Kp values gives us skin permeability. Then PGP substrate, yes, these are PGP substrate. It means they will be metabolized. So they will be transported easily through this substrate, and human intestinal absorption is very good. Then cytochrome P450 inhibitors, no, they are not cytochrome P450 inhibitors. It means that they are not affecting metabolism. Then blood-brain barrier permeant, they are not blood-brain barrier permeant. So it means they will not cross the blood-brain barrier. They will not move to the brain. And then they are non-mutagenic. They are non-human etheragogo related gene potassium ion channel inhibitor. So they are not HERG potassium ion channel inhibitor. And here is the LD50 value, which is lethal dose 50 milligram per kg, and according to lethal dose LD50, which is mg per kg. According to this data, the compounds are slightly toxic. Compound 24 and 34 is somewhat more toxic than 2057. So the lethal dose is the dose which gives 50% death, and the dose in milligram per kg which gives 50% death. So on LD50 we will make another video, the toxicity of the compounds, which is due to mutagenicity, urge potassium ion channel inhibition, or blood-brain barrier, or LD50. So all these are the parameters for their toxicity values. So in this way we get ADMET profile from different softwares and drug-like properties of our compound. Okay, this is our research work that was during my PhD studies, and the research work was published into our 3 reputed international journals. So if you like my video, please comment and share, and also subscribe my channel. Thanks for watching. Allah Hafiz.